the way, I severely updated the slides. So if you've downloaded the slides from the other day, please go back into EduCat and download, because these are really different than when I had a good chunk of it anyways, okay? So we were talking last time with the human microbe. You know, we got through, I think, this slide where we were, that's weird. Oh, there. Whoops. Where we were looking at the idea that we really are a walking ecosystem. We are very complex and have many, many organisms hanging out with us that make us who we are. And so even though, you know, a sperm and egg united to create you, you still have all these other members of the different phyla of life that coexist with you long term. And as we're starting to realize, are doing really important things for you. So if you think about it, when you were conceived and developing in the uterus, they have had a lot of debate about this. What is it like? What is the uterine environment like for the fetus? Is there, you know, any indication that microbes could be there? Now they know already that certain pathogens can make it across the placental barrier and get to the developing fetus. That's been known for a while, but the question is, what about the good microbes, so to speak? Are they present? This has been a really hot debate. It continues. And so some recent studies have been published showing that there are diverse populations, but at low levels, not a lot. Okay, so it's not like there's a lot, but they published studies on the placenta tissue having microbes present, the amniotic fluid, which you're surrounded with as you're a developing fetus. And then remember too, that developing fetuses do swallow amniotic fluid. So that could be a way for the fetus to get exposure internally to microbes that might be in that area of the mother's body. So there's been a few published studies just recently saying it's not sterile. Then this is a title from another group of scientists that said, no, that's wrong. You did it wrong. There's something wrong in your method. And they pointed to the idea that we think you contaminated your tissue samples, your amniotic fluid samples when you did this. So just be aware that the debate continues. Some microbiologists who have done these studies firmly think that it's very possible that there are microbes in these portions of the mother's body, and others are kind of following the old dogma that no, it's sterile. Then another group came along and they said, well, okay, we found what seems to be extracellular vesicles, which remember now, those are coming from a microbe. They create these vesicles and then they kind of pinch off from the cell membrane and go off away from the microbe. And so they found this going on. And so this was published just in 2023. So this idea that there could be microbes that are producing these extracellular vesicles, this is kind of a summary of that paper. And so they're saying, yep, maybe this is going on. It seems like, you know, they are claiming they understand that this is controversial. Recently, extracellular vesicles secreted by the human gut microbiota have emerged as a novel mechanism for host microbiota interaction. So they were looking at the fetal environment in particular and seeing if during a healthy pregnancy, so not pathogens involved, but just a healthy pregnancy, could this be a mechanism? And so they said, yep, we were able to see extracellular vesicles in the amniotic fluid of healthy pregnant women. And so exhibiting similarities to extracellular vesicles found in the maternal gut. And they also did mice studies where they could show that as well. And so this group was saying, well, okay, yes, the debate goes on regarding whether the microbes are there, but we're finding good evidence of the extracellular vesicles. So that's another way if you think about it, that the developing fetus could be getting exposure to microbial stuff, and that would be something important in the fetus's development potentially. And so this paper, this next one, this one was published in the journal Nature, which is one of the most prestigious journals out there. And this was the title, Baby's First Bacteria. The womb was thought to be sterile. Some scientists argue it's where the microbiome begins. And so I won't ask you to read the whole paper. Here is some of the key statements in the paper. And so this first paragraph is against the idea that no, this scientist is saying there are no microbes in there. We would have seen it by now. And he points to the idea that when you work with germ-free mice, they actually take baby mice, surgically remove them, probably, like a C-section from the pregnant mouse mother. And the pregnant mother, it's colonized with microbes. Then we surgically go in and take the baby mice out, and then we put them in germ-free states. He said, if those baby mice had been exposed in the amniotic fluid or the placenta with microbes, we couldn't keep them germ-free. When we do that transfer to a germ-free environment, we see the microbes popping up. So that's his reasoning for thinking that there aren't microbes in the amniotic fluid or the placenta. And then another microbiologist is disagreeing with that and saying that, we have evidence of it. There have been some published studies, even though it's, some people are saying, oh, that's a contamination. So he says in any emerging field, you'll find some naysayers, some dirty data, so like the contamination issue, but also a lot of compelling new observations. So, and that's true. You would tend to get people who will very quickly push back against something that's very new and different from what has been the previous thought for potentially many, many years. So just be aware that this is a debate that's still very current and still have some research to do. I tend to fall into the category that I think it's totally reasonable that there could be low levels of beneficial microbes or the vesicles in the placenta or the amniotic fluid, but we'll see how that progresses. We just need more data. We need more, especially from human studies that we can find and get some good data. Okay, so whether or not that is happening is up for debate, but during the birthing process, as soon as the mother goes into labor and starts into the birthing process, you definitely are going to get the exposure to the baby then at that point. And so during normal vaginal delivery, of course, the vaginal microbiome will colonize the baby, and especially lactobacillus species. And if you look on any, like if you look at yogurt or probiotics that they sell, lactobacillus is a very common genus that you see with many different species in that genus. They have seen that if the vaginal microbiome has decreased community richness, so the overall population in the vagina tissue, the microbes just the populations aren't high, you don't have as much diversity. They actually have been correlating that to preterm birth, that it can cause issues and including preterm birth. So they're actually screening mothers. Right now it's in the trial phases where they're screening mothers, pregnant mothers, to see if they can come up with kind of a marker of being able to discern this baby is at risk of a preterm birth. And so live biotherapeutics is kind of their strategy. They're like, if we can detect that the community richness is low, we could augment that microbiome, the vaginal microbiome, with live microbes, sometime during the pregnancy in order to help with that. So they are looking at ways to help with preterm birth issues. So as the labor is continuing, you have colonization from microbes that are in the vaginal tract, the feces, of course. Labor is a very physical process. It's not clean and tidy. So feces, certainly, skin, breast tissue, and breast milk. And so they've also had controversy, although I think it's fairly well accepted now, that even breast milk is colonized with microbes as well as the breast tissue. And so the baby is getting microbial exposure that way too. And over again, they show it really does depend on the environment too that that baby is surrounded in. You know, when you look at different families, like are there pets in the house? Are there other children in the house? What is that baby getting exposed to in the very, very early days of its life? And so definitely they can show over and over that the surrounding environment is very impactful for the microbiome development. So of course, this issue has come up. What about babies that are born via C-section? They're not going through the vaginal tract, so they're not getting that exposure that, you know, and when you think about humans and other animals, the vaginal birthing method is the natural way. That C-sections came about fairly recently, and it's very unnatural, but sometimes very, very necessary. So when you think about it, the microbes that are in the vaginal canal are coexisting long-term with that female. And if you think about a C-section where they cut open the stomach and then they get to the uterus. So now the baby is going to get exposed primarily to skin bacteria. That'll be its first exposure. And skin bacteria, whether it's the mom's skin or the nurse and doctors and all the other people in the hospital as they are working with the baby, they're going to get that. And skin microbes are inherently more ephemeral. They don't persist. They're not always the ones that coexist long term. There's a lot more fluctuation in the types of microbes that are found on the skin surfaces compared to internal tissue like the vaginal tract. And so this idea that the baby's immediately getting exposed to chance microbes and not long-term coexisting microbes is something that they point to that could be something of concern and definitely could impact the baby. And so some studies have shown that there's increased risk of chronic inflammatory diseases with babies that are born via C-section. And so they're still, of course, doing a lot of research on this, but they've noted a increased risk of asthma and diabetes and celiac disease. So that's an important question to ask. If you don't know how you were born, you might want to ask about that process. And then the other idea of, okay, once the baby's born, oh, by the way, going back to vaginal birth versus C-section, there are currently, within the last handful of years, there's been a push from mothers that are aware of this idea that if they have to do a C-section, they want the baby to be swabbed with vaginal microbes from their vaginal tract. And it's interesting how that has met with really a lot of doctors, especially the surgeons, are really against it, which is they always point to, what if there's pathogens in the vaginal tract? And it's like, well, yeah, of course you screen for that. It's like, I don't know why there's such a resistance, but I've talked to several women who have been scheduled for C-sections because of medical issues. And they wanted to do, you know, a normal birth, but they were told they'll have to do a C-section. And then when they asked their doctor, you know, can I have the baby swab? You know, they had to really fight for it. So just be aware if that's something any of you end up doing at some point, whether you're a woman or, you know, you're married to somebody who's going to give birth. Think about this idea of, you know, if it's a C-section, you may want to consider that. They can easily screen for pathogens in the vaginal tract. So it's not insurmountable. And in my opinion, I think it's well worth at least considering that idea. But just be aware you're probably get met with some resistance on that. Okay, and then this has been a topic too of debate. What about breastfed versus, when I say bottle fed, I mean formula fed. I guess that would have been a little bit more precise. Breastfed versus formula fed is more what I'm thinking, because you can certainly pump and put breast milk into a bottle. That's not really what I'm thinking about. So if you think about that, when they track this through different cohorts of developing babies, they can find if the baby has been breastfed for about 6 months, that they seem to have greater microbial resiliency in their microbiome. And then the breast milk itself has its own microbiome. There are microbes that are in the breast milk long-term, as well as the breast tissue. And so they get exposure that way. And then if you look at breast milk, it contains what they call human milk oligosaccharides. HMO is how they abbreviate it, human milk oligosaccharides. So oligosaccharides, oligo means like shortish chains of sugars. So it's not a polysaccharide, it's an oligosaccharide. And so what they found, okay, so they're present. They're like, okay, the breast milk has this. What, is it feeding the baby? And they were shocked to realize that even though it's a large component of the breast milk, it doesn't directly feed the baby. It can't be digested by babies. They don't have the correct enzymes for it. So they're like, oh, okay, what's going on here? So the human breast milk is designed to feed the microbes that are developing in the baby and in the baby's gut. I mean, that is just fantastic. It's just amazing. And so bifidobacterium in particular, they see with breastfed children, babies, that their bifidobacterium levels really rise to good levels. And one of the papers I read, they put in there, bifidobacterium benefits babies, BBB. And so bifidobacterium, that genus of bacteria, what it can do is it can ferment pyruvate to lactate. And when they do that, they ferment in a way that doesn't produce gas. Now, if you know of anyone, or if you are a mom and had a child that has colic, it is not fun. My younger son had that, and having that gas buildup in the intestinal tract is very painful. It's very slow to go away. They cry and cry and cry. They're very uncomfortable. And so it is definitely something you want to avoid. You get like zero sleep for many days on end if it persists. So that is just one benefit that they point to with having a good level of bifidobacterium. And then they're also finding that it can produce anti-inflammatory compounds. And we're going to talk a lot about inflammation in this section of the course, because that is a huge topic right now when they look at human microbiome issues. And bifidobacterium also produces beneficial immunomodulators, which means it's helping the baby's body get its immune system up and running and getting it working appropriately rather than inappropriately. So that's just one example of how the human milk oligosaccharides are benefiting the different microbes in the baby's microbiome. The other idea, this paper was just published not that long ago, a couple years. So they're saying formula feeding, they're showing, is creating a situation where the microbes that occur under those conditions actually exhibit more antibiotic resistance, which obviously you don't want. And of course, remember now, when you think about the gut environment, if you have microbes with antibiotic resistance, don't forget their ability to do horizontal gene transfer and get that transferred to other members in the microbiome in the gut. And so this is of real concern. They also found, I should say, here they're saying a higher relative abundance of opportunistic pathogens, such as Staph aureus, Staph epi, Klebsiella pneumoniae, oxytocin, all of those can be opportunistic. And then ARGs are antibiotic resistance genes. They also saw those 69% higher in this particular study compared to the controls that were fed human milk. So another reason to seriously consider doing the breast milk idea, at least for the initial, they say 6 months for sure is a good idea. Some say longer than that, up to a year. Okay. Here is another paper. This one was just published last year. So again, looking at breastfed versus formula fed. And this was what they call a meta-analysis where they looked at published studies. And so they went into different search engines and did analyses of different published papers, and they were trying to draw conclusions from, all of these different papers. And so from that, they worked with 13 different publications that they did their analysis on. And they could show that, again, breastfed had higher levels of beneficial, like the bifidobacterium we already talked about, lactobacillus, while the formula fed had more prevalence of pathogens present.
We're talking about the breastfed versus formula at their house. And one of the things that they brought up is that sometimes it's hard to do these studies on the children because it's hard to have a control group. And that, like, one of the ideas that they brought up was that, like mothers who breastfeed their children might be more inclined to do healthier habits because they are breastfeeding.
Yeah.
Do you think that, like, that would have any effect on the microbiome of the baby, or do you think that it would be more because of the breast? Do you think that would play a factor in it or not?
I think it could play a factor. I'm hoping that when they set up the study, they're careful to discuss these ideas with people participating and trying to find participants that have a more similar way of interacting with their baby, I guess. I think it could have an impact. But I think overall, the strongest impact is from what they get fed. And especially if it's young babies where they feed so frequently, so they're getting a pretty steady exposure to breast milk or formula. And so it's kind of swamps out some of the other factors, I think, as they get.
I'm just curious.
Yeah, I mean, that's a really good, and you're right, it's hard to find humans to participate in a lot of this, because some of it you just can't do in a human, it's too invasive. And then there's all these other variables that can mess with your data and how you think about it, so. Yeah, it's always, that's why a lot of it's mice, where they control everything really tightly. And so. But eventually you gotta get into the human, at least for some of these stuff. Eventually you have to start testing against, with the human as the participant. So, and when you think about it, even as adults, when you interact with your environment, your food choice is probably your most intimate interaction with your environment, right? Because that's something that you take into your body. It's not just, like when you take a shower, it just washes over you. But when you eat food, it interacts very intimately with all these different cells and organs in your body. And so we'll talk a good bit about that too, about how food can impact so highly what's going on with the microbes in you. So here's a schematic from that paper. Just looking at, what's going on in terms of, I didn't really mention yet that we do see immune cells in the breast milk too, so that the baby's getting exposed to important immune cells right early on. So it's not just the nutrients, of course, but also some of the immune cells and enzymes and other factors. And so I know that a lot of formula-fed companies are trying very hard to mimic what the breast milk contains. As far as I know, no one's perfected that, but they have gotten better than what, a few decades ago was available. One other study that I was looking at was showing that bottle-fed or formula-fed babies seem to have higher levels of microbes that can degrade mucus. And I'm going to be talking more about mucus. So this is actually a negative thing in some regard, because if you have too much of this going on, you weaken the barrier between your body and the microbes especially in your intestinal tract. And so you want a layer, actually there's like two distinct layers of mucus especially in your colon area, which has the highest amount of microbes compared to like your small intestine. And so I'll come back to this idea of it's okay to have some microbes that do this, that's fine. And actually some of them are very beneficial, but if you get too many of these doing this, it can be weakening the barrier. So this interest in the microbiome, how does it develop from, the onset of labor through, they usually cite the first 3 years of life is pretty important and a really critical time period for the microbiome to develop. And so in general, what it seems to be showing, many published studies are showing that as the infant is developing, you eventually get to a point where they say it's now more like the adult microbial profile. And basically, you get certain microbes starting to dominate over others. It's kind of like when you learned in ecology about a forest successional processes, how some of the tree species come in first and then they get out competed by some of the other tree species that come in second. So you have this kind of successional process going on in the microbiome as well. And these 4 tend to dominate when you get into adult life. And I would like you to know these 4 names, please, and whether or not they're gram-negative and gram-positive, because that will factor into some of our discussions. So the Bacteroidetes, people will pronounce these differently. So this is just my pronunciation. You probably hear it differently in other settings. Bacteroidetes are gram-negative. Firmicutes are gram-positive. The proteobacteria are gram-negative, and the actinobacteria are gram-positive. Now, other studies are tacking some additional phyla onto this list, but when I go back historically from the start of human microbiome research to current, these 4 sets still are very dominant in the adult microbiome. But having said that, we are all unique. Every single person has a unique microbiome because these are groups, these are phyla names, all right? And then within these phyla, each of these phyla, there's so many different species. And so your life experiences, your diet, all of that factors in. So your microbiome is as unique as your fingerprint. All right, everyone has a unique microbiome because when you get down to the species level, there's going to be differences. And so a lot of microbiologists say this is not a random process. It is an exquisitely orchestrated program. I really like that quote. And so again, it really does depend on your behavior and activities and what you interact with, how your microbiome is going to stabilize, who's going to be there. And you actually have a lot of say, so to speak, in that. You have a lot of control on that in many ways, and we'll talk about that. So here is from a paper, and again, going out to the first 3 years of life. And so they're just trying to give a feel for how things start to shift at some key steps in this first 3 years. And so obviously, when you go from just milk to a mixed diet of milk and food, that's a huge change. And so again, that will be a major break where you're going to see some differences showing up depending on what the baby gets fed, which, there's a lot of food choices out there. And so that you're going to start to see some major shifting going on. the other thing that you see too is once you start introducing solid food, you get increased species diversity, which makes sense. You know, it's no longer such a, milk, it's milk plus a lot of different foods. And so of course, it's going to be increasing the diversity. So with some of the adult studies that they've done, this is just trying to get you to think about the idea that you can have some stability. So they're saying, 60 to 70% remained unchanged over the course of the study. But you can have some microbes that are more sensitive to perturbation. So if there was a change in diet, for example. And so it really does depend on what the people are doing, how some of the nuances of the microbiome are going to change over time. But they do, I mean, after a couple of decades now of this kind of research, they're still saying, yeah, the adult does have a fairly stable profile at the phyla level. But when you get down into the species level, we are all very unique. Yes.
Yes, in the adult microbiome.
Yes.
I heard that people in the same household and even like pets can have similar microbiome.
Yes.
And is that like strictly due to diet or are there other indirect interactions?
Part of it's diet, but like I know some families where, someone's a vegan and someone's a carnivore, and they still are, not that's showing differences. And then other families where they all eat the same and they're still different, so it varies, but mostly what they point to is, and over the studies show one household, they'll have more similar microbes compared to the neighbors right next door. And so a lot of that is just what's in their house, what's in the air when they touch things and it transfers to their body. So you have a lot of the environmental influence, including food. So that's one reason why I felt so good when I learned about the idea that, babies should be exposed to dirt and so it's like, good, I don't have to clean my house as bad as I thought, as much as I thought I did. Because once you have babies, it's like, well, you get worried about them getting sick. When I found out this information, I'm like, good, I'm gonna leave some of those dust bunnies and some of the dirt around, and I didn't worry about it. So, and they never got sick from that kind of stuff. So anyways, this paper was published just last year. And so they're trying to develop this well-being index for the gut microbiota. And again, they're trying to think about if we can learn enough about the human microbiome, then we could step in and do some preventative health or more aggressive therapy early on. And so here they were trying to track 1,000 infants, which that's a really good sample size. And so they were trying to see, can we, by studying these 1,000 infants and their microbiomes, can we come up with, this is a healthy microbial profile, and then use that as a diagnostic tool for trying to assess if, a child is under risk. And so they are seriously thinking about this. And now that there's more and more data out there, it's getting a little easier to consider this as possible. And so where is it? We characterize the natural healthy gut microbiota trajectory and several dysbiotic, we'll talk about dysbiosis in this class too. And they're trying to use that. So they're saying, could we figure this out so it's very predictable? We understand that this profile means this is going on and the child is at risk for this. And so it's very promising. I think when in your generation, and if you choose to have kids, that generation too, this will be coming into play more and more. And so it's very encouraging to see that scientists are thinking this way. And they couldn't really do that, just like a decade ago. There wasn't enough data out there to really start thinking about it in that regard. So the first 3 years of life in particular, and So really what you want happening is you want the developing baby, the toddler, to get exposure to diverse microbes. And one huge reason for that is that it does help train the immune system. And they have shown this in mouse studies where they take germ-free mice and compare them to conventional mice that have a microbiome. Germ-free mice don't have a microbiome and they're raised in totally sterile conditions. And they look at the immune system, vastly different. The immune system in the germ-free mice is way underdeveloped. Even their intestinal tract is shorter and different. And we're going to learn, if you don't know already, that a good chunk of our immune system is in our gut. It's where a huge part of our immune system is. And so the microbes need to be there in order to train the immune system. And so you got to kind of train the immune system to say, okay, now's the appropriate time to start inflammation, to fight. But they also have to learn, okay, but these microbes are okay. They're not going to hurt you, so don't fight against them. I mean, it's a delicate balance because your immune system has to figure out who's friend, who's foe, and then what is an appropriate response to deal with the problem. You don't want autoimmunity kicking in, obviously. And so again, many scientists have published papers showing they think all of this is going on during the first 1,000 years, or 1,000 days of the baby's life. And so, getting your immune system up and running. I don't know if anyone's taken immunology where you might talk about this idea, but I'm guessing Dr. Sharp would say the same thing with his immunology training that he has. Okay. So very important that you do, don't worry about a super clean house. That's not necessary. In fact, is actually can be detrimental to the developing baby. Having good exposure and diverse exposure to different environments is considered to be very good. And so when you start thinking about microbe exposure, we are, at least for most of what we're talking about in this class, for this section, we're talking about the resident microbes, all right? And so when you say that term, you mean the ones that are coexisting with us long-term over our lifespan. And so when I'm talking about the human microbiome, that's mostly what I'm focusing on. The transients are the ones that are present, but usually for shorter time periods, anywhere from hours to maybe a few months, but then they fade away. And then the opportunists are like, the opportunistic pathogens. If the conditions arise that make it so it's easy for them to start to attack and get access to more nutrients, then we start to call them opportunists. And so whether that be from the host being stressed out and their immune system goes down, or they get sick with something else, and so then they start to get an upper hand, could be from surgery if you have a traumatic accident even, or a surgery that can introduce pathogens into, or just microbes into places of the body that they shouldn't be in. And so then we start saying opportunists. So you could see like Staph aureus on the skin is totally fine, not the MRSA type. Do you know what MRSA means? MRSA is the bad type, methicillin resistant Staph aureus. So you don't want that, but just normal Staph aureus on your skin, that's fine. But if it gets into your internal body, like through if your skin wasn't disinfected and you had surgery, or you got into a car accident and some of the microbes got into the wound, then Staph aureus could become opportunists. So just an opportunist isn't always evil. Yes. It depends, like, how did you get it in the first place? Like, is there something you're doing in your daily life, or something like, there's a study done with wrestlers, and that competitive wrestlers were getting higher rates of Staph aureus infections just from, because of other wrestlers having it. And then they even tested like the wrestling mats and could find like after competitions, the presence of staph, MRSA. And so these types of ideas. Okay. Well, let's look at the skin. There's been research on the skin. Of course, that's our immediate barrier to the environment. You want a good protective barrier. You want good, healthy skin. You should be drinking good hydration, including electrolytes, not having a lot of gashes and wounds, of course. And I like this figure taken from a published study because it's with the pie charts, you're seeing a lot of different colors depending on what's part of the body. This isn't just the skin, it's looking at other parts too, but as you can see, depending on the habitat of the human, you're going to see differences. And that kind of makes sense, right? That when you look at these different habitats, they're different environments for the microbes that they're experiencing. And so, this is just a nice visual getting you to think about kind of breaking down the body into different habitats that have unique physical and chemical properties that different microbes find, ideal and others not so much. And so you get these differences. Remember too, if you haven't had any anatomy for a while, that your skin has many layers to it, right? So the epidermis is on the very outside and then the dermis and the subcutaneous area. And we do have like hair follicles and sebaceous glands that provide sites for the microbes to hang out. It's a nice protected site. And in particular, the sweat glands and sebaceous glands are producing chemicals that a lot of microbes find nutritious. So it's a way for them to stay well fed. Sometimes the sweat can be, because of the saltiness, it does make a lot of microbes not want to be there. It's too salty. And so you get this selective environment in this case with higher salt concentration. But if you can, if you just look at the hair follicle, for example, you can imagine that, yeah, microbes could get down in there, no problem. They exist in a more sheltered environment. So when I say skin, yes, they're on the epidermis, but they're down into the dermal layers. And then remember, you do shed your epidermis on a regular cycle. And so it makes sense that, you can always swab your skin and get microbes showing up. They're always there. The only way you could get rid of microbes on your skin is if you burn that section of your skin. Obviously, you don't want to do that. You want the good microbes on your skin because then they help outcompete the transients that come in that might be opportunistic. Of course, some microbes can cause problems with skin and cause inflammation and infections. That's we've all experienced that over our lives. And so I like this one too, nice color coded here showing these. So this is specific to the skin. And again, you're seeing different areas, is it dry skin? So certain parts of your body are drier, like your forearm is more dry than your back or your chest, for example. And so moist skin would be maybe your back. And then the sebaceous skin, you have oilier areas on your face. In other areas. And then, even like thinking between your toes, that's a very protected environment. But anyways, you can see here that a lot of what's showing up, 3/4 of the pie chart here is gram positives. So gram positives are very much well suited for the skin environment. Gram negatives, they have a little tougher time. There's some specialists, but fewer in number compared to the gram positive. Cool. Well, let's go. Oh, we're out of time. It's 2:50 already. All right, so we'll stop there and we will continue on. We're going to kind of look through different habitats of the human body. And then eventually we'll get more into the gut microbiome. Have a good weekend. Be careful. It's going to be icy and snowy. Be really, really careful.
You think burn wounds